There is something almost embarrassing about how long it took us, not decades, centuries. And across those centuries, in anatomy theatres from Bologna to Edinburgh to Paris, rooms filled with the smell of tallow candles and something else, something sharper that no one talked about. The same thing kept happening. A body would be laid out, a blade would descend, and the first layer encountered, the pale, membranous, fibrous stuff that appeared before anything recognizable did, would be peeled back and set aside. It wasn't dramatic. That's the part worth sitting with. There was no decision made, no meeting convened, no collective agreement reached across continents that this particular tissue was unimportant. It was simpler than that. The tissue just kept getting in the way of things that looked more like something, and so it was moved, cut around, discarded into whatever vessel sat at the edge of the table for precisely that purpose. Welcome to the Sleepy Biologist. I'm glad you're here. The channel exists because of the people who choose to support it, those who've become members, who send a super thanks, who leave a small quiet note in the comments. You probably know who you are. And if tonight is your first time here, welcome into this particular kind of darkness, which I hope is comfortable. For centuries, the tissue had no fixed name. Different anatomists called it different things: paniculus, common membrane, fibrous envelope. Nothing stuck. And in a way, that namelessness was itself the problem. A thing without a name is harder to argue for. It is harder to point to in a diagram and say, this matters, stop throwing it away. The naming came slowly, unevenly, in fragments. The word fascia, from the Latin for band, for bundle, for something that wraps, eventually settled into use. But even with a name, the attitude didn't shift quickly. Muscle had fibers you could see contracting. Bone had density, structure, the visible logic of a frame. Nerve carried electricity. Blood moved. These things announced themselves. Fascia did not announce itself. It sat there, white and slightly glossy, yielding to the blade without complaint. It connected things, but the things it connected were what everyone came to see. It enclosed everything. But enclosure isn't glamorous. You don't build a career on the casing, you build it on what's inside. So it was set aside chapter after chapter of human anatomical history. And what that means, what that quiet dismissal actually cost us, is only becoming clear now in the last few decades, when the instruments got finer and the questions got stranger, and researchers started looking at what had always been there. I find something a little melancholy about it. Not tragic, just melancholy. The way you might feel looking at an old photograph and noticing someone at the edge of the frame slightly blurred who turned out to be important. The tissue that was discarded is now understood to be a system, not a single structure, not a passive wrapping, a continuous three-dimensional network that laces through every part of you, beneath your sweat, between your muscles, around your organs, along your nerves. It doesn't begin somewhere and end somewhere else. It simply continues. If you could somehow remove everything that wasn't fascia, you would be left with something that still held your shape. That is not a small thing, and yet it was scooped aside for centuries in rooms that smelled of candles and cold. Part of what makes this strange is that the surgeons weren't careless people. The anatomists weren't lazy. They were precise, obsessive even, the kind of people who notice things. They just weren't noticing this particular thing, because nothing in their training had told them it was worth noticing. And training is powerful that way. It tells you very quietly where to look. Everything outside that frame can become invisible, not through malice, but through habit. There's something we still don't fully understand about how long this can persist, the overlooking of something essential. It's not a failure of intelligence, it seems more like a failure of category. The tissue didn't fit neatly into what anatomy at that time needed things to be. It wasn't an organ, it wasn't a muscle group, it wasn't a system, in the way the cardiovascular system is a system. With a clear direction, a clear purpose, a clear beginning, you can point to on a diagram. It was connective. That word contains the problem. Connective tissue connects, and connection maybe was simply not interesting enough. Until it was. The shift, when it began, didn't come from a single discovery. There was no one moment, no paper that changed everything overnight. It came gradually, the way understanding usually comes, through accumulation, through researchers in different countries arriving at overlapping conclusions, through a first international congress on facial research in 2007 that apparently surprised its organizers by attracting far more attendees than expected. People have been quietly thinking about this for years. What they were beginning to realize, what is still being realized in laboratories and anatomy departments right now, while you're lying here, is that the tissue nobody wanted had been doing something all along. Not just holding organs in place, not just providing padding, something more active, more specific, more involved in the ordinary running of the body than anyone had guessed. Your fascia right now, as you lie still, is not inert. It is full of cells, a particular kind of cell whose job is to sense when a layer of tissue is under strain, another kind whose job is to produce the lubricant that keeps your internal layers sliding past one another without friction, a third kind that has only recently been given a name at all, found by researchers at the University of Padua a little over a decade ago, different enough from everything around it that it had to be classified as new. These are quiet workers. They don't announce themselves. But then nothing about this tissue has ever announced itself. That may be the thing that strikes me most, returning to it. Not the complexity, not the recent discoveries, but the patience of it. The way this system spent centuries being set aside, cut around, discarded, unnamed, and simply continued doing what it had always done. The body didn't wait for us to notice. It just kept working. And somewhere in the space between your skin and the structures beneath it, something thin and continuous and very, very old is exactly that right now. Something has been happening in your body for longer than you can remember having a body. Not a dramatic process, not one that announces itself in sensation or shows up in the mirror. It's been running the way certain background things run, the way your liver filters overnight, the way your corneas repair their outermost layer while you sleep, quietly and without any particular audience. Fascia does this. The cells inside it have been manufacturing, adjusting and rebuilding the tissue they live in for your entire life, and they were doing it before you were born, and they were doing it right now while you lie here. I find that strange to hold on to, actually, that the cells in a tissue most of us have never consciously thought about are, right now, engaged in something that looks, at a cellular level, like labour. The predominant workers are called fibroblasts. The name is dry and functional, which suits them. Fibro for fibre, blast for making. They make fibre. Specifically, they make collagen, the most abundant protein in the human body, the structural backbone of skin and tendon and bone, and the dense connective sheeting of fascia. Fibroblasts synthesize collagen in particular types, type 1 for strength, type 3 for suppleness, with the proportions varying by location and by what the tissue has recently been asked to do. But fibroblasts aren't only building, they're also listening. This is the part that seems to get underappreciated when people talk about connective tissue, the fact that these cells respond. Not slowly, not passively. When a fibroblast experiences mechanical strain, when the tissue around it is pulled or compressed or sheared, it changes its behavior within minutes. It spreads. It alters the expression of genes inside it. It adjusts the composition of the extracellular matrix, that intricate gel and fiber environment in which every fascia cell is embedded. The cell is, in a real and measurable sense, reading the mechanical situation of your body and responding to what it finds. You've probably felt the downstream effects of this without knowing what to call them. The gradual stiffening of tissue that has been held in one position for too long. The slow release that comes from sustained movement, from warmth, from something pressing gently and persistently into a resistant area. That's not just muscle. Some of what you're feeling is connective tissue adjusting, fibroblasts registering a change in load and beginning quietly to alter what they produce. Outside the fibroblasts, suspended in the extracellular matrix, are adipocytes, fat cells that aren't simply energy reserves stored passively away, but structural participants. They occupy the loose connective tissue within facial layers, softening the architecture, providing what a material scientist might call compliance, the quality of yielding without breaking. Between them and around them flows the ground substance, a gel composed largely of glycosaminoglycans and hyaluronic acid and water, so much water held in a mesh of molecules that can absorb up to 1,000 times their own weight in fluid. Not metaphorically, literally. The extracellular matrix of fascial tissue is one of the most hydrophilic environments in the body. And then there are the cells discovered more recently, macrophages doing immune surveillance inside fascial layers. Mast cells, identified in the superficial fascia of the abdomen only in 2023, their presence suggesting an immune sensing role that no one had mapped before. Telecytes, a cell type found in the fascia lata and thoracolumbar fascia, and a handful of other fascial structures whose precise job remains genuinely unclear. They're there. They've been counted. What exactly they're doing is still being worked out. I think about that sometimes. The cells whose purpose we've confirmed but can't yet fully explain. There's something grounding about it. The body has always been ahead of our descriptions of it. The fascia site is worth pausing on. It was found in 2011 by researchers at the University of Padua who were doing detailed histological work on fascial tissue, the kind of slow, meticulous looking that requires cutting samples into sections 5 micrometers thick and staining them and examining them under high magnification. They found a cell that looked somewhat like a fibroblast but wasn't quite. Plumper, different in what it expressed, different in where it lived, positioned specifically at the interfaces between fascial layers and the surface of adjacent muscles. Its function, when they worked it out, turned out to be the production of hyaluronic acid. Not everywhere, but exactly where hyaluronic acid was most needed, at the boundaries where one layer of tissue must slide past another. It had been there all along, in every body, doing a specific necessary job. No one had named it. The community of cells inside fascia, then, is not a simple one. It's not one cell type doing one thing in one place. It's a diverse, locally organized population of cells that communicate, respond, build, degrade, and rebuild. And that has been doing this, in your body specifically, since sometime in the first trimester of your fetal development, when the mesoderm folded and differentiated and began laying down the first sheets of connective tissue that would eventually become the continuous network you carry now. That continuity is the part I keep returning to, not the complexity of it, not the biology, but the sheer uninterrupted duration. Your fascia has not paused. It has not taken a break while you slept, or while you were ill, or during the years when you were growing fast and nothing felt quite right in your own body. The cells have been working through all of it, adjusting, replacing degraded collagen fibers, producing lubricant, sensing load. There's no fanfare in this, no signal you receive. You don't feel the fibroblasts synthesizing collagen in the thoracolumbar fascia while you shift in bed. You don't feel the extracellular matrix absorbing fluid, or a fasciocyte releasing hyaluronic acid at the interface between your iliotibial band and the muscle beneath it. These things happen below the threshold of anything your nervous system is designed to surface, but they are happening. Right now in the tissue most anatomists once cut away without a second thought. A slow and ancient and completely ordinary form of construction is underway. The same work that was running while you were a child, while you were an adolescent, while you were doing things you remember and things you've forgotten. The same work that will keep running tonight in the dark without any instruction from you. The body doesn't need you to know about it. It never has. And yet here you are, knowing, or beginning to, and the cells inside you are indifferent to that knowledge, continuing exactly as they were, measuring load and producing fiber and holding the whole complicated arrangement in a state that is never finished, never quite complete, always slightly in the process of becoming whatever it needs to be next. You have felt this before without knowing what it was, the particular resistance of a body that has been still for too long. Not pain exactly, more like the tissue itself has forgotten that moving was an option. You shift and something doesn't quite shift with you. You stretch in the morning and there's a drag, a low, stubborn protest from somewhere between muscle and skin that takes a few minutes to release, if it releases at all. Most people assume this is muscle, tight muscles, dehydrated muscles, something in the muscle. It often isn't muscle. Between your fascial layers, between the dense fibrous sheets that wrap your muscles and the surfaces of the muscles themselves, there is a substance that is neither solid nor liquid, and that has one primary job: to stop those surfaces from catching on each other. It does this by being wet. That's the simple version. The longer version involves a molecule called hyaluronic acid, which is not an acid in any way you'd recognize from the word, but a long looping chain of sugars, technically a glycosaminoglycan, that exists throughout your body in synovial joints, in the aqueous humor of your eyes, in the umbilical cord, in wound sites, and most relevantly here, in the loose connective tissue that separates fascial layers from each other and from the muscles they enclose. What hyaluronic acid does, structurally, is hold water, not a little. A single hyaluronic acid molecule can bind up to 1,000 times its own weight in water. That number deserves a moment, not because it's dramatic, but because it explains something. Why the space between fascial layers feels, when things are working correctly, almost frictionless. Why tissue that should be sliding does slide. The molecule creates a hydrated gel in the interfascial space, a slick film that allows one layer to move independently of the layer beside it. Without it, the surfaces would drag. With too little of it, or with the wrong kind, they already do. Outside, right now, there might be the faint sound of something, a fan cycling on, a car passing on a wet road, the particular silence that follows. The body next to that sound, your body, has hyaluronic acid in dozens of locations, quietly doing this work. The cells that produce it in fascial tissue are called fasciocytes, discovered formally only about 15 years ago. They sit at the margins, at the interfaces between fascial layers and muscle surfaces, and they secrete hyaluronic acid into exactly the space where it's needed most. What triggers them is movement, shearing motion. The mechanical action of one surface sliding past another stimulates the fascia site to produce more of what makes that sliding possible. This is the part that stops me a little. The lubricant is produced by the act of lubricating. Movement generates the signal for more of what movement requires. It's not a loop exactly, but it rhymes with one. And what breaks it, what interrupts it, is stillness. When tissue is immobilized, hyaluronic acid production continues, but its recycling slows. The molecule accumulates. At higher concentrations, something changes in its behavior. It begins to aggregate, to clump, its long chains tangling around each other in ways that reduce its water-binding capacity. The gel that was thin and slick becomes thicker, more viscous, less lubricating. The layers stop sliding. What happens then? In tissue that has been held in the same position for too long, whether by injury or immobilization or simply by the way you sleep or sit or carry tension, is called densification. The fascial layers adhere, not catastrophically, not all at once. Gradually, through a slow accumulation of thickened gel and reduced movement, the surfaces that were designed to glide begin instead to drag against each other. The nociceptive nerve endings threaded through that tissue register the change in mechanical tension, and somewhere in the upstream processing of that signal you feel it as stiffness, or as pain, or as that particular morning heaviness that doesn't quite fit the word stiffness, and doesn't quite fit the word pain, but lives in the territory between them, in the low back, in the hips, in the soles of the feet when they first meet the floor. There is a strange comfort, I think, in knowing this. Not because knowing it makes it better, it doesn't, immediately, but because it moves the thing out of the category of mystery. The stiffness is not arbitrary. It's not your body malfunctioning. It's a gel that has thickened in response to specific conditions, doing exactly what a thickened gel does, which is resist. And resistance, under the right conditions, can be reversed. What reverses it is also movement, but specific kinds. Mathematical models published in 2013 looked at how different manual therapy motions affect the flow of hyaluronic acid in fascial tissue. They found that rapid oscillating pressure, a back and forth motion perpendicular to the tissue surface, produced far higher rates of hyaluronic acid redistribution than slow linear strokes. The gel under oscillation disperses. The aggregated chains separate. Water binding capacity returns. The layers gradually begin to slide again. Your body has a system for lubricating itself that depends on being used. Not aggressively, not dramatically, used. Moved through its ranges regularly in ways that produce the shearing motion the fascia site is listening for. When that happens, it secretes. When it secretes, the gel maintains its quality. When the gel maintains its quality, the layers slide. When the layers slide, you move without the drag you might have assumed was simply age or tension, or the particular character of your body on a given morning. It might not be any of those things. It might be a molecule, thickened slightly from sitting still, starting to thin again now that you've moved, or starting to thin tonight while you sleep as the body shifts position and the tissue responds to even that small mechanical event, the way it always has, without instruction, without awareness on your part, in the dark space between one layer and the next, where a gel that can hold 1,000 times its weight in water is either flowing freely or resisting, and either way, you'll feel it in the morning when you move. Close your eyes, though they may already be closed, and think about what your body knows right now, or what you saw, what your body knows, the angle of your neck against whatever is beneath it. the slight compression of your shoulder or your hip, or the outside edge of your foot if it's pressing into the mattress at an odd angle, the weight of the blanket, the temperature differential between the parts of you that are covered and the parts that aren't. Your body is receiving all of this information simultaneously, continuously, and routing most of it away from consciousness entirely, because none of it requires a decision, it's just data, incoming, processed below the threshold of attention. A significant portion of that incoming data is generated by fascia, 250 million. That's the estimated number of nerve endings in the human fascial network. The number was pulled together by the researcher Robert Schleip, whose work on fascial neurology helped shift how the field understood what this tissue was actually doing. 250 million. Your skin has around 200 million. Your eyes contain approximately 126 million photoreceptors, and yet the tissue that was routinely discarded in anatomy theatres for 5 centuries turns out to hold more sensory apparatus than either of those. The air in the room right now has the temperature. You probably stopped noticing it an hour ago. That forgetting, that routing of sensation away from active attention, is exactly what the fascial sensory system is built for. Not to alert you, to maintain you, to run a continuous background accounting of where your body is, what loads it's carrying, what tensions have been building, what positions are becoming unsustainable. The information doesn't arrive as a message. It arrives as a postural adjustment you didn't decide to make, a shift in muscle activation you weren't aware of, a change in the way you're distributing weight that happened without any involvement from the part of your brain that uses language. The nerve endings themselves come in different kinds, and the differences matter. The encapsulated receptors, Ruffini endings, Pacinian corpuscles, Golgi organs, are found most densely in fascial tissue near joints and in the extremities, near the ankle, under the arch of the foot, in the tissue surrounding the wrist. Ruffini endings respond to sustained stretch and lateral tension. Pacinian corpuscles attune to vibration and rapid changes in pressure. Golgi organs, more commonly associated with tendons, exist in fascial tissue too, registering the kind of slow cumulative tension that builds over time. These are the receptors that handle what researchers call proprioception, the body's continuous internal mapping of its own position in space. You've felt proprioception fail, briefly and probably only in small ways. The strange, slightly vertiginous moment when you misjudge a step in the dark. The fractional lag between your foot meeting an unexpected surface and your leg correcting for it. That lag, that small window where the system is catching up, is the proprioceptive apparatus updating its model. Most of the time it's fast enough that you never notice the gap. When it isn't, you notice it very suddenly. The fastest nerve fibers in the fascial system are the mechanoreceptors. They carry urgency. When you almost fall, they're part of what fires before any conscious awareness of falling has occurred, before the word falling has even begun to form. The signal travels, the muscles respond, the correction happens, and then, a fraction of a second later, your heart rate spikes, because the cognitive part of you has just caught up to what the sensory part of you already handled. But the majority of nerve endings in fascia are not encapsulated. They are free nerve endings, bare, unencapsulated, threaded through the collagen of fascial layers in dense branching networks. What they detect is harder to categorize. Mechanical strain, chemical changes in the local environment, temperature to some degree. Some of them are nociceptors, pain receptors, which explains why fascial pain, when it occurs, can be so diffuse and so difficult to locate precisely. The signal isn't coming from a joint or a muscle belly. It's coming from a distributed network embedded in the wrapping of everything, and it presents as a sensation that doesn't have clear edges. Some of these free nerve endings appear to serve interoception, the sensing of internal states, heart rate, digestive movement, the vague embodied awareness of how you feel, not emotionally but physically, from the inside. The research here is less settled. As best we can tell, some of the sensory apparatus in visceral fascia, the connective tissue around internal organs, participates in this quiet inward monitoring. The gut feeling that is actually a feeling registered in tissue, not invented by imagination. What seems to be the case when you pull back and look at what's been mapped so far is that fascia constitutes something close to a body-wide sensing surface, not a brain, not a nervous system in the conventional sense, but a distributed sensory organ, one that operates at every scale simultaneously, from the crude and urgent balance, impact, threat, to the slow and subtle accumulated postural strain, sustained compression, the gradual drying out of a gel between layers. Carla Stecco, the orthopedic surgeon and anatomist at the University of Padua, whose work on fascial structure has been foundational to the field, has described the thoracolumbar fascia, the broad diamond-shaped sheet of connective tissue across the lower back, as something like a large receiver. Tension arriving from the arms, the legs, the spine, the abdominal wall, all of it is registered there, not interpreted in any way that generates conscious awareness, just registered, filed, incorporated into the body's running model of itself. The model is never static. Right now, as you breathe slowly, probably, if you've been lying here for a while, the fascial tissue across your ribcage is being gently loaded and unloaded with each cycle. The free nerve endings there are registering this. The Ruffini endings in the connective tissue around your ribs are tracking the sustained stretch at the top of each inhale. None of this surfaces as sensation because it doesn't need to. Your body already knows what breath feels like. The data confirms what the model expected, and so it passes through without flagging anything. This is most of what sensory processing is. Confirmation, expected inputs matched against existing models, quietly filed. It's only the unexpected that gets escalated. The step in the dark, the uneven surface, the sudden load on a joint that wasn't braced for it. And in those moments, the fascial sensory system is among the first to respond, even though it rarely receives credit for doing so. What happens when this system becomes less accurate? When fascial densification or injury or chronic inflammation alters the mechanical environment of those nerve endings, changes the tension around them, shifts the baseline that the receptors are calibrating against, is poorly understood and probably underestimated. The proprioceptive signal becomes less reliable. The body's model of itself drifts from reality. Compensation patterns emerge in muscles that weren't designed for the loads they're now carrying. The observer with preferences in me wants to say this is the most consequential part, not the structural role of fascia, not even its role in pain, but this, its role in the accuracy of the body's map of itself. When the map is wrong, everything downstream adjusts to fit the wrong map. But that's a big thought for the quiet end of a chapter. What's simpler and maybe more useful to carry into sleep is just this. Beneath your skin, woven through the tissue that connects everything to everything else, 250 million nerve endings are at work right now. Not sleeping, not pausing, measuring, reporting, holding open the channels through which your body continues to know itself, in the dark, without your help. They have been doing this since before you knew they existed. They'll still be doing it when you wake. A wound doesn't know what to do with itself. This sounds almost absurd, but it's the right way to think about it. The moment tissue is damaged, whether by a cut, a tear, a burn, the controlled incision of a surgical blade, the site of injury becomes a place of tremendous chemical noise. Signals fire, vessels respond, fluid rushes in. The local environment shifts its chemistry within seconds in ways that are measurable and specific, and yet, at the level of the wound itself, undirected. The wound is a problem that has been declared. It is not on its own a solution. Something else provides the solution, or rather, something else provides the instruction. For a long time, the question of where the instruction came from was genuinely open. Researchers knew that fibroblasts arrived at wound sites and began depositing extracellular matrix. They knew that certain fibroblasts became myofibroblasts, contractile cells that pull wound edges together and build the scaffolding of new tissue. They could observe the sequence of events. What they couldn't fully trace was the origin. Where did these cells come from? What determined that the right cell type arrived at the right moment? Why did the sequence unfold in the order it did? The answer, when it came, was not where most people expected to find it. It was in the fascia, specifically in a subpopulation of cells sitting in the deepest connective tissue layer of the skin, not the dermis, not the muscle, but the fascial layer beneath both, that carry on their surface a protein marker called CD201. The discovery was published in November 2023 in Nature from researchers at Helmholtz Munich, and it reframed something that had seemed, until then, like a downstream process into something that looked unmistakably like orchestration. CD201 positive cells are progenitors. That word matters. A progenitor is not a finished cell. It is a cell that retains the capacity to become multiple different things, to differentiate in response to the right signals into specialized cell types that it was not before. CD201 positive fascia progenitors, when a wound occurs in nearby tissue, do exactly this. They generate the cells that wound healing requires, and they do so in a sequence that is spatio-temporally precise. The inflammatory fibroblasts arrive first. The myofibroblasts come later. Each type appears in a timed window that corresponds to the phase of healing that needs it. Not randomly, not all at once, in order. The signal that drives the first transition, from resting progenitor to pro-inflammatory fibroblast, is retinoic acid. You may know it from skincare. Here its role is different and older and more fundamental. It is a chemical checkpoint, a gate. When retinoic acid levels are high in the wound environment, the progenitor cells tip toward the inflammatory state, producing chemokines that recruit immune cells, amplifying the initial response that clears debris and begins preparing the site. The retinoic acid also, critically, prevents those cells from becoming myofibroblasts too soon. It holds the sequence in place. Then retinoic acid levels fall, and a different signal rises. Hypoxia. The wound environment becomes locally oxygen depleted. Deeper regions of the wound, farther from the surface, receive less oxygen as the normal vascular supply is disrupted. Hypoxia-inducible factor 1 alpha, HIF1 alpha, responds to this. And under its influence, the CD201 positive progenitors tip into their second transition, becoming myofibroblasts, the contractile matrix-depositing cells that physically close the wound and lay down the structural material of scar tissue. Two signals, two transitions, timed by the chemistry of the wound itself. When the researchers genetically ablated the CD201 positive lineage in mice, removed it specifically, while leaving the rest of the cellular architecture intact, wound healing didn't simply slow down. It stalled. The timed sequence of fibroblast types failed to appear. The wound remained open chronically in a way that ordinary variation in healing never produced. The lineage wasn't one contributor among several. It was load-bearing. Outside, somewhere, there is probably the sound of something cooling, a house settling, the particular quality of silence that comes after a certain hour. Your body is doing none of what it would be doing if you were wounded. But somewhere in your fascial tissue, the CD201 positive progenitors are present. Waiting is the wrong word. They aren't waiting for anything. They are simply residing, maintained in a state of readiness that requires no active effort on their part or yours. The 2019 Nature paper from the same research group had already suggested something related, that when deep wounds occur, fascia physically mobilizes, not metaphorically. Fibroblasts from the fascial layer migrate toward the wound site, moving through tissue to reach the place where repair is needed. The fascia is not a static reservoir. It moves toward the problem. I find it difficult to know exactly what to do with this, intellectually. Not the biology is clear enough, but the implication that beneath the layer most anatomists once threw away, there is a population of cells whose specific purpose is to manage the aftermath of damage, that the tissue dismissed as packaging contains the repair system for everything inside the package. It was there in every body that was ever dissected and discarded without a second look. In every anatomy theatre, in every surgical suite, in every specimen that was cut through to reach the muscle or the organ or the nerve. The cells that coordinate healing were in the white membranous layer that hit the floor first. The wound healing cascade that plays out now in any body after any injury at all begins not at the wound but just outside it, in the fascia, in a cell that carries a surface protein and holds the capacity to become whatever the moment requires if the signals arrive in the right order, which they usually do, because the signals are themselves generated by the conditions of the wound, which means the wound is, in a strange and recursive way, partially responsible for its own repair. Not because it knows anything, because the system was built that way, and the system was built or evolved over timescales that make the history of human anatomy look brief, in fascial tissue, in the wrapping, in the layer that was for so long the first thing the knife moved aside. The cells are still there, right now, in the fascial layer beneath your skin, in the same tissue that encases your muscles and anchors your organs and holds threaded through it 250 million nerve endings. The progenitors are present. The machinery is intact. Nothing is injured. Probably the cells don't need anything to be injured to exist. They are simply there in the wrapping as they have always been, ready for something that may never come or may come tomorrow, doing what progenitor cells do in the absence of a signal, which is very nearly nothing, which is its own kind of readiness, quiet and cellular. And very, very old. Think about the last time you moved quickly and unexpectedly, not exercise, as something more ordinary than that, catching something that was falling, stepping off a curb that was slightly lower than you anticipated, twisting to grab a door before it closed. There is a quality to that kind of movement that feels different from deliberate motion. It happens before you've decided to do it. The body has already responded before the mind has fully registered that responding was required. And the response is not just one muscle contracting. It is a coordinated event across multiple structures simultaneously, a redistribution of force through whatever tissues happen to be connecting the moving parts at that moment. Some of that redistribution happens through fascia, the thoracolumbar fascia, the broad layered sheet that spans the lower back from roughly the shoulder blades to the sacrum, receives mechanical input from the arms, the legs, the spine, and the wall of the abdomen simultaneously. Carla Stecco has described it as a large receiver, which is accurate but perhaps undersells the directionality of the exchange. It doesn't just receive, it transmits. A contraction in the latissimus dorsi generates tension that travels into the thoracolumbar fascia, and from there into structures the contracting muscle never directly touched. This transmission is not incidental. It is how the body distributes load across regions wider than any single muscle can span. The term for this, epimuscular myofascial force transmission, was formalized by the Dutch biomechanist Peter Huijing in the early 2000s, though the phenomenon itself had been observable in cadaveric studies long before anyone named it. Huijing demonstrated that muscles transmit force not only through their tendons, the conventional pathway every anatomy class teaches, but through the connective tissue surrounding them, to adjacent muscles, to bones, to fascial sheets nowhere near the origin or insertion of the contracting muscle. The mechanical reach of a single contraction turned out to be substantially wider than the muscle's own boundaries. There's something almost relieving about this, I find. It explains things that the single muscle model never quite could. Why stretching one area sometimes releases tension in an area that seems unrelated. Why a restriction in the plantar fascia, the thick band along the sole of the foot, can, through the continuity of the superficial backline of connected fascial tissue, influence the mechanics of the hamstrings, the thoracolumbar fascia, even the cervical region. These connections have been demonstrated with ultrasound imaging. Move the pelvis in a specific way, and the fascial tissue around the gastrocnemius shifts cranially in a measurable, reproducible displacement. This is not metaphor. Ultrasound captured it. The body workers who'd been saying something like this for decades, that the body is continuous, that tension in one region communicates with tension in another, had, it turned out, been observing something real. The science arrived late to a party the hands had already attended. The implications for pain are significant and still being worked through. Somewhere in a room right now, probably many rooms, a person is being told their back pain has no identifiable cause. The imaging is clean, the discs are intact, the vertebrae are aligned, and yet the pain persists, diffuse and stubborn, in the lower back, and nothing in the standard diagnostic framework explains it. This is not a rare situation. Somewhere around 90% of chronic low back pain cases are classified as non-specific, meaning the underlying structural cause is not identifiable with conventional imaging. Helene Langevin, a researcher who has spent decades studying connective tissue and is now director of the National Center for Complementary and Integrative Health in the United States, began in the early 2000s to measure fascial mobility in people with and without chronic back pain. What her group found in a 2011 study using ultrasound elastography, a technique that measures how tissue deforms under shear force, was a 20% reduction in the shear strain of the thoracolumbar fascia in people with chronic low back pain compared to pain-free controls, 20% less movement in the fascia. The imaging showed no structural damage. What it showed was a change in mechanical behavior. The layers of the thoracolumbar fascia were moving less relative to each other than they should. The tissue was not broken, it was restricted, and the nerve endings threaded through that restricted tissue were registering the altered tension in their environment as pain, a diffuse, poorly localized, difficult to treat pain that had been attributed, in the absence of better explanation, to muscles, to posture, to stress, to aging. The quality of fascial pain is distinct from muscle pain when researchers have been able to isolate and compare them. Fascial pain tends toward burning, cutting, sometimes stinging, a quality different from the dull ache of muscle soreness. The difference matters clinically, because it suggests the pain is not coming from where most treatment is directed. Outside, it might be raining now or perfectly still. Your lower back, wherever it is making contact with the surface beneath you, is probably not in pain right now. If it is, you know exactly what I mean? If it isn't, there's probably a memory nearby, a morning, a long drive, a day at a desk, where something in that region felt heavy and uncooperative in a way that no amount of shifting position quite resolved. That may have been fascial. What's less understood, and this is genuinely unresolved, is why the restriction persists in some people and not others. Why the same loading pattern produces densification in one person's thoracolumbar fascia and apparently glides through another's without consequence. Inflammation plays a role, hyaluronic acid chemistry plays a role, movement history plays a role, but the specific interaction between these factors and why the restriction sometimes becomes self-sustaining, even after the original provocative load is removed, is not yet fully mapped. The body, in this as in most things, is more complicated than the models built to describe it. What is clear is that the fascial network, the continuous sheet of connective tissue that connects your plantar fascia to your hamstrings, to your thoracolumbar region, to the connective tissue around your shoulders and neck, is a single communicating system. Tension in one region is not isolated. It propagates. It influences adjacent structures, sometimes distant ones. The body manages this propagation continuously, redistributing load, adjusting tension, absorbing through the viscoelastic properties of fascial tissue what cannot be redirected. It does this while you stand, while you sit, while you sleep, shifting position, each shift a small mechanical event that ripples outward through the network, loading and unloading and reloading structures in a slow, dark, wordless conversation that your body has been having with itself for every hour of your life, and will keep having quietly through the rest of this one. The sections were 5 micrometers thick. To cut tissue that thin, you embed it first, in paraffin usually, and use a machine called a microtome, which advances the specimen in increments so small they have to be calibrated. Then you stain the sections because unstained biological tissue is largely transparent, and different stains bind to different structures and render them visible in different colors. Then you look, for a long time under magnification, at what the staining has revealed. This is the work. It is slow. It requires the kind of attention that doesn't announce itself as remarkable, because it's just what the work requires. Somewhere in a room that probably smelled faintly of reagent and warm equipment, researchers at the University of Padua were doing exactly this in 2011. They were examining fascial tissue, the deep connective tissue that wraps and separates muscles, and they found something that didn't quite fit the categories they'd brought with them. A cell, but not the fibroblast they expected. Plumper than a fibroblast, rounder. Different in its surface markers, which is how cell biologists distinguish between types, by what proteins the cell displays on its outer membrane, the molecular flags that identify function and lineage. This cell was positive for vimentin, a marker shared with fibroblasts, but it was negative for CD68, which ruled out the monocyte macrophage lineage. It sat in a specific location, not scattered throughout the facial tissue, but positioned deliberately at the interfaces between facial layers and the epimysium, the surface of the underlying muscle. It lived exactly where the two structures needed to slide past each other, and it was producing hyaluronic acid, large amounts of it. The staining made this clear. Alcyon blue, which binds to the acidic polysaccharides that hyaluronic acid belongs to, showed concentrated production in this cell type, production confirmed by immunohistochemistry, and then by real-time PCR analysis that detected strong expression of HAS2, the gene encoding hyaluronic acid synthase 2. The cell was making the lubricant, at the exact location where lubrication was most needed. It had a name by 2018 when the follow-up paper was published in Clinical Anatomy. Fasciacyte, from fascia, the tissue it inhabited, and cyte, from the Greek for cell, a straightforward name for something that had been present in every human body ever studied and had never been formally described. That gap between presence and description is worth sitting with for a moment. The fasciacyte wasn't hiding. It wasn't rare. It wasn't a pathological finding or a variant. It was a normal, abundant, functionally essential component of fascial tissue that simply hadn't been identified because no one had been looking for it specifically, or looking at the right locations, or asking the question that would have required finding it. The question being, what cell exactly is producing the hyaluronic acid in this particular interfascial space, and is it distinct from the fibroblasts nearby? Someone had to ask that. Then someone had to look. Then someone had to sit with section 5 micrometers thick and stain them and look for long enough that the difference became visible. Outside, the dark is very quiet, the kind of quiet that accumulates after a certain hour, when the ambient sounds of movement and activity have dropped away and what remains is just the base level of wherever you are. In your fascia right now, fasciacytes are doing what they do, no signal required, no injury prompting them. They are producing hyaluronic acid at the interfaces between your fascial layers and the surfaces of your muscles, in your thighs, your back, your shoulders, the connective tissue of your arms, maintaining the hydrated film that allows those surfaces to slide. They were doing this before anyone had named them. They did it through every surgery in which the fascia they lived in was cut through and set aside. They did it through the decades in which the tissue itself was classified as structurally unimportant. They did it without any recognition of what they were, because they are not cells that require recognition. They just make what the sliding needs, and the sliding continues. It begins before you notice it. A few hours into immobility, a limb in a cast, a joint that hurts too much to move, a long convalescence in bed. The fascial tissue surrounding the affected region begins to change. Not dramatically. Not in ways that would show up on imaging, not yet. But at the level of the extracellular matrix, things are already shifting. The hyaluronic acid that normally maintains the lubricated film between fascial layers continues to be produced, but its clearance slows. The molecule accumulates. As concentration rises, its behavior changes. Long chains begin to tangle and aggregate, losing water binding capacity, thickening from something that allows surfaces to glide into something that makes them drag. This takes hours, not days. Hours. The tissue isn't damaged yet. The cells aren't dying. The collagen architecture is intact. But something in the mechanical environment of the fascia has already begun to drift from its normal state. And the drift, if stillness continues, compounds. By the time days have passed, the changes are more structural. Fibroblasts, the cells responsible for maintaining the collagen framework of fascial tissue, are mechanosensitive. They respond to load. When load is absent or dramatically reduced, the signals that would normally regulate collagen production and degradation fall out of balance. The ratio shifts. New collagen is laid down in orientations that suit a static tissue rather than a moving one. Fibers crossing at angles that provide stability for a position held, rather than the multi-directional arrangement that permits a range of movement. The tissue begins to reorganize around the fact of its own stillness. There is something almost logical about this, from the body's perspective. resources are not wasted. If a joint is not moving, the connective tissue surrounding it adapts to the mechanical reality it's actually experiencing, not the one it might experience if things were different. The adaptation is not malicious. It is metabolically efficient. The body is doing what bodies do, adjusting to the environment it finds itself in, conserving what it can, reinforcing what seems to be in use. The problem is that the adaptation outlasts the reason for it. A joint immobilized for 6 weeks changes its fascial architecture in ways that don't simply reverse when the cast comes off. The crosslinks between collagen fibers, chemical bonds that form between adjacent strands and affect how much the tissue can stretch and recoil, have multiplied during the period of immobility. Cross-linking is a normal part of fascial maintenance, giving the tissue strength and coherence. But excessive cross-linking, the kind that accumulates when collagen is being deposited without the remodeling that movement stimulates, produces a stiffer tissue, one that resists elongation, that transmits force less smoothly, that moves less freely relative to adjacent structures. The stiffness you feel after weeks in a cast is not entirely the stiffness of disused muscle. Some of it is the collagen architecture of the surrounding fascia, which has been quietly reorganizing itself into something optimized for staying still. Outside, the night is at whatever hour it's reached. There may be the low sound of the building adjusting and pipes, or the particular creak of a structure cooling. Small sounds, the kind you only hear when everything else has gone quiet. What happens over months, in bedridden patients, in long recoveries, in joints that are guarded and offloaded because they hurt, is more significant still. The fascial layers that should slide past each other begin to adhere, not through scar tissue initially, but through the thickened hyaluronic acid gel and the cross-link collagen, and the absence of the mechanical forces that would normally keep the surfaces separate. Adhesions form. The loose connective tissue that provides interfascial glide consolidates into something denser, less differentiated. And what follows is the thing clinicians call restricted range of motion, not because the joint itself is damaged sometimes, but because the connective tissue surrounding it has reorganized itself around an architecture of immobility. The myofibroblasts are part of this story too. These contractile cells, the same cells that close wounds, can become overactive in fascial tissue under certain chronic conditions. They generate tension independent of neural input. A fibroblast that has tipped into a myofibroblast state in response to sustained low-grade inflammation or mechanical to coil stress doesn't simply relax when the provocative condition resolves. It can maintain its contractile state, sustaining tension in the fascial tissue around it, contributing to this environment of restriction. This is the condition called adhesive capsulitis, frozen shoulder, in one of its proposed mechanisms, not primarily a joint pathology, a fascial one. The connective tissue of the joint capsule, populated by activated myofibroblasts and reorganized by immobility, has become something that restricts the joint it was meant to support. I want to be careful here not to overstate what's known. The precise threshold between restorative rest and damaging immobility, how many hours, at what level of restricted movement, in which tissues, in which people, is genuinely not mapped with the kind of precision that would let you calculate it. The biology is clear in direction. It is less clear in dose. What is clear is that movement is not optional for fascial health in the way it might be optional for, say, a bone that can maintain its architecture under relatively low mechanical demand. Fascia is a tissue that requires the input of movement to maintain the fluid dynamics and cellular signaling and mechanical diversity that keep it functional. Without that input, it adapts. And the adaptation, as adaptations often are, is very hard to mistake for anything other than what it is, the body making the best of what it has been given. The tissue carries this history differently than the mind does. You might not remember 6 weeks in a cast when you were 19, or 3 months of reduced activity during a long illness, or years of a posture that loaded one side more than the other. The fascia may still hold some record of it, not as damage necessarily, but as altered architecture. As collagen laid down at angles that suited a different mechanical reality. As subtle differences in how layers glide, or don't, in a particular region. This isn't catastrophe, it's history. Biological history written in the extracellular matrix, available to the tissue but not to the conscious mind. And tonight, as you shift, even slightly, even the small unconscious repositioning that happens through the night without waking you, the fascial tissue responds. The layers slide fractionally against each other. The mechanical input reaches the fibroblasts and the fasciocytes. The hyaluronic acid, whatever its current state, experiences the shearing motion that stimulates production and turnover. The body is not entirely still even now. It is making the small, continuous adjustments of a living thing, and those adjustments, however modest, are something the fascia reads as movement, as load, as the ongoing evidence that this tissue is still in use, still worth maintaining, still part of a body that is going somewhere, even if that somewhere is only deeper into sleep. 90%. That's approximately the proportion of chronic low back pain cases classified as non-specific, meaning no identifiable structural cause, no herniated disc pressing on a nerve root, no fracture, no tumour, no measurable spinal pathology that would explain what the patient is describing. The imaging is reviewed. The report comes back unremarkable, and the person sitting across from the clinician, who has had pain in their lower back for months or years, and whose daily life has reorganized itself around that pain, is told, in various phrasings that amount to the same thing, "We cannot find what is wrong." This is not a small number of people. Low back pain is among the most prevalent disabling conditions globally. It costs healthcare systems and individuals and economies sums that are difficult to hold in the mind, and the dominant diagnostic response to the majority of cases has been, for decades, a category called non-specific, which sounds scientific, as categories do, but is functionally a record of what was not found rather than what was. I find non-specific a strange word to live with, if you're the patient. What researchers began to suspect, gradually and then with increasing evidence, was that the imaging was looking in the wrong places. MRI and CT scanning are exceptionally good at revealing structural pathology, bone, disc, nerve compression. They are not designed to measure the mechanical behavior of soft tissue layers relative to each other. And so the thing that was actually different in many chronic low back pain patients, not the structure of the spine but the mobility of the connective tissue surrounding it, was simply invisible to the tools being used. Helene Langevin had been studying connective tissue for years when her group turned to this question specifically. She was already known for work on how acupuncture needle rotation mechanically engages fascial tissue, work that had been received with some skepticism, which is perhaps unsurprising, but which was methodologically careful, and whose findings about fibroblast response to mechanical stimulation were real and replicable. When she began applying ultrasound elastography to the thoracolumbar fascia of people with and without chronic low back pain, she was asking a simple question with a non-simple answer. Do the fascial layers move differently in people who hurt? The answer, published in 2011, was yes. Ultrasound elastography measures shear strain, the degree to which a tissue deforms when a shearing force is applied. In people without chronic low back pain, the thoracolumbar fascia shears normally during passive spinal flexion. The layers slide against each other as the spine bends, displacing relative to one another in a measurable, reproducible way. In people with chronic low back pain of more than 12 months duration, this shear strain was reduced by approximately 20%, 20% less movement, in the fascia, not in the disc, not in the vertebra, in the layered connective tissue that wraps the musculature of the lower back. And in men specifically, there was a positive correlation between the duration of pain and the degree of reduced shear strain. The longer the pain had been present, the less the fascia moved, which suggests, though causality is difficult to establish, either that restricted fascial mobility produces chronic pain over time, or that chronic pain produces progressive fascial restriction, or both, cycling. What explains the pain if the structure is intact? The thoracolumbar fascia is densely innervated. This has been established in multiple studies. Free nerve endings, nociceptors, the full sensory apparatus discussed in an earlier chapter of this particular journey, through what the body is made of and how it runs, when facial layers are restricted, when the hyaluronic acid has thickened, when collagen has reorganized around immobility, when the interfascial glide that the tissue depends on is reduced. The mechanical environment of those nerve endings changes. They are embedded in tissue that is under different tension than it should be. That altered tension is a stimulus, and stimulus in a nociceptor is registered, eventually and with some processing, as pain. The specific chemistry involved has been partially mapped. Nerve growth factor, produced by inflamed tissues, sensitizes nociceptors, lowers their threshold, makes them fire more readily in response to stimuli that wouldn't normally reach the level of pain. Studies in rats demonstrated that an inflamed thoracolumbar fascia shows a measurable increase in nociceptive fiber density compared to intact fascial tissue. The tissue grows more sensors in response to sustained irritation, which is not a system designed with comfort in mind, but with detection, with making sure the signal keeps being sent. Calcitonin gene-related peptide and substance P neuropeptides released by nociceptive fibers in fascial tissue have been measured at elevated levels in and around the thoracolumbar fascia in chronic pain states. When released centrally, these peptides contribute to sensitization at the level of the spinal cord, what's called central sensitization, where the pain signal is amplified not just locally but in the neural processing itself. The pain becomes, in a sense, self-sustaining. The peripheral signal and the central processing reinforce each other. This is a digression worth making because it explains something that confuses people about chronic pain, why it doesn't always respond proportionally to changes in the peripheral tissue. You can treat the fascia, improve its mobility, reduce local inflammation, and the pain may persist because the central sensitization has become its own phenomenon. The system learned to amplify. Relearning to quiet it takes different work. Outside, the night is at its lowest point, the part of it where the air feels thinnest and the silence is most complete. Your back, wherever it is making contact with whatever is beneath you, is probably somewhere between these two states, neither perfectly mobile nor genuinely restricted. Most people exist in that middle ground. Some portion of morning stiffness, some occasional ache, some direction of movement that requires a little more negotiation than the others. The fascial tissue of the lower back is thickened in people with chronic pain consistently across imaging studies. It is less compliant. The collagen content increases. The myofibroblast activity in some cases is elevated, contributing to sustained intrinsic tension that doesn't depend on muscle contraction and doesn't resolve with muscle relaxation. The tissue has, gradually and without intent, become different from what it was before the pain began. Or possibly it was already becoming different before the pain became conscious, the structural change preceding the symptom, the restriction present before the complaint. Which came first is genuinely unclear in many cases. There is a particular discomfort in that uncertainty, especially from the perspective of someone who has spent months or years being told their back pain has no identifiable cause. What the research offers is not a complete answer. It offers a plausible, measurable, increasingly well-documented mechanism that the standard diagnostic toolkit was never designed to find. The pain was not imagined. It was not purely psychological. It was not a sign of weakness or of exaggerated sensitivity. It was, in many cases, a signal from connective tissue that had changed its mechanical behavior in ways that activated the sensory apparatus embedded in it. That the tools to see this clearly enough took so long to develop is simply the history of medicine, which advances unevenly, which sees what it has the instruments to see, which sometimes requires decades to look in the direction that the patients were pointing toward all along. The thoracolumbar fascia of a person with chronic low back pain is not broken. It is not diseased in the way an organ becomes diseased. It is altered. Its layers less mobile, its chemistry shifted, its nerve endings running in an environment of different mechanical tension than they were built for. Their body is not failing. It is responding persistently and somewhat miserably to a set of conditions that have become self-reinforcing. Understanding the mechanism doesn't dissolve the pain. It doesn't restore the shear strain to its previous value or quiet the sensitized nociceptors overnight. But something about having a name for what is happening, a real name, a measurable name, a name that points to an actual physical process in actual tissue and not to the absence of anything findable, seems to me at least like it should matter, like it belongs to the person in pain, as information they were owed from the beginning. The fascia of your lower back is doing something right now, probably something unremarkable, the ordinary maintenance of a tissue in a body that is resting. The layers are wherever they are relative to each other. The hyaluronic acid is whatever viscosity it has arrived at tonight. The nociceptors are quiet, or nearly so. But the system is there. It has always been there. And it has been for a very long time, talking. In the only language connective tissue has, which is tension and load and the chemistry of a gel that can hold 1,000 times its weight in water, or can thicken slowly into something that no longer quite lets the body move the way the body was made to. There is a version of getting older that happens in the mirror, and a version that happens somewhere underneath it. The mirror version is familiar. The gradual changes in skin, in posture, in the effort required to recover from physical demands that used to cost less. These things are visible or felt, and they accumulate in ways the mind can track. But the connective tissue version of aging, the changes happening in the fascial network, in the collagen architecture, in the molecular environment between layers of tissue, runs on a different timeline, deeper, earlier, below the threshold of anything that would flag itself as a change until, suddenly, something is noticeably less than it was. The body's facial aging begins, in some measurable senses, before most people think of themselves as aging at all. Collagen is at the center of this. The most abundant protein in the body, the structural backbone of fascial tissue, exists in your fascia as a dynamic material, being produced, degraded, and replaced in a process called collagen turnover. In younger adult tissue, this turnover is relatively brisk. New collagen is synthesized, degraded collagen is cleared, and the fiber architecture remains organized in a way that balances strength with elasticity. The fascia can stretch and recoil, it can absorb load across a range of directions and recover. What changes with age is partly the rate of this turnover, and partly what the collagen looks like when it's replaced. Collagen fibers are not isolated strands. They are organized into fibrils and then into bundles, and the bundles are held together partly by crosslinks, chemical bonds that form between adjacent collagen molecules, binding them to each other and contributing to the structural integrity of the fiber. Some crosslinks are normal and necessary, the kind that form rapidly as new collagen matures and that give the fiber its tensile strength. Others form more slowly over years, through a non-enzymatic process called glycation. Glucose molecules reacting with collagen proteins and producing what are called advanced glycation end products, which accumulate in long-lived connective tissue over time. These accumulated crosslinks make the fiber stiffer, not weaker necessarily, stiffer. The collagen bundles become less forgiving, less able to deform smoothly under load and return to their original configuration. The tissue loses some of what materials scientists call viscoelastic compliance, the capacity to absorb and distribute force by temporarily deforming. A fascia with high crosslink density bears load differently than one with lower density. It transmits force with less modulation, less dampening. The mechanical signal arrives at the fibroblasts and nerve endings with fewer of the qualities smoothed out. And then there is the question of fiber organization. Imaging studies have shown that with age, the collagen in fascial tissue becomes less regularly organized. Fibers laid down at subtly different angles, the clean parallel architecture of younger tissue giving way to something slightly more disorganized, more tangled in places. The intramuscular connective tissue thickens. The interfascial glide, the capacity of fascial layers to slide past each other, mediated by the hyaluronic acid gel maintained by fasciocytes, becomes less smooth as the layers become thicker and less differentiated. Somewhere outside there is the particular stillness of a night that has fully settled. No particular sound, just the background of wherever this is. The sex hormone dimension of fascial aging is less often discussed, and probably less often understood than the collagen story, but it is real and specific. Fascial tissue carries receptors for estrogen. Fibroblasts within the fascia respond to estrogen signaling by modulating their production of collagen types 1 and 3 and a structural protein called fibrinogen, which contributes to the elastic properties of connective tissue. When estrogen levels fall, as they do during perimenopause and menopause, this signaling is reduced and the balance of collagen production shifts. Type 1 collagen, which is stiffer and less elastic, becomes relatively more abundant. Type 3, which is more pliable, decreases proportionally. The fascia, in other words, gets stiffer not only because of accumulated crosslinks and fiber disorganization, but because the hormonal environment that was partly regulating its composition has changed. The two processes are independent and additive. This may be part of why the subjective experience of flexibility loss often accelerates noticeably in the perimenopausal period, not because muscle has suddenly shortened dramatically, but because the fascial architecture is changing its properties in response to a shift in hormonal signal that it had been organized around for decades. The tissue was built in a particular endocrine context. When that context changes, the tissue changes with it. The relationship between testosterone and fascial properties is less clearly mapped, but there are differences in fascial thickness and stiffness between males and females that are documented in imaging studies. Differences that shift again across the lifespan in ways that suggest sex hormones are not peripheral to fascial biology but integrated into it. What this means practically is that the stiffness many people experience as they move through middle age, the morning negotiation with the body, the longer warm-up required before movement feels fluid, the sense that recovery from physical effort takes more than it used to, is not purely a muscular phenomenon. It is not purely a joint phenomenon. Some of it is the fascia, which has been changing layer by layer, fibre by fibre, cross-link by cross-link, since early adulthood, at a pace slow enough to be invisible until it isn't. There is something I find quietly disorienting about this. Not distressing, but just strange. The idea that the tissue has been moving in this direction for years before you noticed. That the body you inhabited in your 20s had fascial tissue that was already beginning the long process of becoming what it is now. That the changes visible in function today are in some sense already underway when nothing felt different yet. I'm not sure what to do with that exactly. The body doesn't offer a warning. There's no signal that says the cross-link accumulation has passed some threshold, no notification that the interfascial glide has reduced by a meaningful percentage. It just changes. And at some point, different for different people, influenced by movement history and hormonal trajectory and genetics and the specific mechanical demands of a particular life, the accumulated change becomes perceptible. The morning stiffness that takes longer to resolve. The position that produces an ache it didn't used to. The range of motion that is simply no longer what it was. What the research consistently shows is that movement history matters significantly to this trajectory. Not in a way that stops the aging, it doesn't, but in a way that changes its pace and its severity. Fascial tissue exposed to regular varied movement retains better organization of collagen fibers. The fascia sites in regularly loaded tissue maintain more active hyaluronic acid production and turnover. The interfascial glide is preserved more effectively in bodies that have kept using the full range of what the fascia was built to support. This is not a prescription. It is just the biology. The tissue rewards use with maintained architecture. It reorganizes around stillness. It has always done both of these things in every body across every life. Tonight your fascial tissue is at whatever stage it is at, crosslinks accumulated to whatever degree, collagen organized with whatever degree of regularity, the interfascial glide whatever it currently is. These facts are not a verdict. They are a snapshot of a long, slow process that began in the first years of your life and will not stop until the last. Right now, though, in this particular hour, the process is barely moving. The fibroblasts are cycling slowly. The fasciocytes are producing hyaluronic acid at the low rate appropriate to a body at rest. The crosslinks are crosslinks, fixed and quiet, doing nothing dramatic in either direction. And somewhere deep in the connective tissue of your back or your shoulders or the soles of your feet, collagen fibers that have been there for years are just sitting in the dark, in the matrix, in the quiet, holding their shape as they always have, as they will for a while longer. And the aging is so slow tonight as to be almost indistinguishable from stillness. A wound is a problem that has been declared but not yet solved. The moment tissue breaks, whether by accident or by the controlled intention of a surgical blade, the local environment shifts its chemistry with extraordinary speed. Vessels constrict, then dilate. Fluid moves in, platelets aggregate. The inflammatory cascade initiates. All of this happens in seconds, and all of it is real and necessary, but none of it is repair. It is the announcement of damage, the clearing of a site, the chemistry of a body recognizing that something has been breached and beginning urgently to do something about it. What it does next is more complicated than it first appears. For decades, the broad shape of wound healing was understood in phases. Inflammation first, the immune response, the debris clearance, the local swelling and warmth and redness of a wound in its first days, then proliferation, the arrival of fibroblasts, the deposition of new extracellular matrix, the scaffolding of repair being built across the wound site, then remodeling, the maturation of scar tissue, the gradual reorganization of collagen into something load-bearing and permanent. 3 phases, sequential, each giving way to the next. What wasn't understood was the origin of the cells doing the work. Fibroblasts arrived at wound sites. That was established. Myofibroblasts, contractile matrix depositing, appeared in the proliferative phase and contracted the wound edges. This was documented. But where these cells came from, what determined that the right type appeared at the right time, what prevented the inflammatory fibroblasts from becoming myofibroblasts too early or too late, these questions were open in ways that the phase model couldn't address, because the phase model described the sequence without identifying the source of the sequencing. The source was in the fascia, specifically in a population of cells identified in 2023 by researchers at Helmholtz Munich, cells carrying a surface marker called CD201, residing in the deepest connective tissue layer beneath the skin, in the fascial tissue that had been the first thing surgical protocols moved aside for a very long time. These cells are progenitors, not finished, not committed to a single fate, but capable, under the right chemical conditions, of differentiating into multiple specialized cell types. What the 2023 Nature paper demonstrated, through genetic lineage tracing and single-cell transcriptomics and carefully designed ablation experiments, was that these CD201 positive fascia progenitors do exactly this, and do it in a sequence so precisely timed that the word choreography, which researchers themselves used, seems almost restrained. The sequence begins when injury signals reach the fascial progenitor population. Retinoic acid rises in the wound environment during the inflammatory phase. This is the first checkpoint, a molecular gate. Under elevated retinoic acid, the CD201 positive progenitors differentiate into pro-inflammatory fibroblasts. These cells upregulate chemokines including CCL2, recruiting monocytes and macrophages to the wound, amplifying the inflammatory response that clears debris and prepares the site for construction. And retinoic acid simultaneously prevents those cells from tipping prematurely into the myofibroblast state, holding the sequence in place, keeping the inflammatory phase inflammatory for as long as it needs to be. Then retinoic acid levels fall, and the wound environment deeper in its tissue becomes hypoxic, oxygen depleted, as the disrupted vasculature fails to supply the inner regions of the wound adequately. Hypoxia-inducible factor 1 alpha rises. HIF1 alpha is the second checkpoint, the second gate. Under its influence, the CD201 positive progenitors tip into their second transition, differentiating into myofibroblasts, the cells that physically contract the wound edges and deposit the extracellular matrix of new tissue. The construction phase begins, 2 signals, 2 gates. The timing determined by the chemistry of the wound itself. There is something recursive in this that the narrator in me wants to sit with for a moment. The fact that the wound environment generates the signals that control the sequence of its own repair. The hypoxia is produced by the disrupted vasculature of the wound. The retinoic acid gradient is shaped by the inflammatory response in the wound's outer layers. The repair system is reading the wound to decide what to do next, and the wound is being produced by the injury, which means the injury is, in a strange and indirect way, informing its own resolution. Not because anything intends this, because the system was built, evolved, over timescales that make human anatomical history seem brief, to read exactly these signals and respond to them in exactly this sequence. Outside, the night is at whatever depth it has reached. Something in the room has a temperature that is easy to stop noticing after a few minutes, and you probably stopped noticing it some time ago. The ablation experiment made the argument definitively. When the researchers genetically eliminated the CD201 positive lineage in mice, specifically this lineage, while leaving the rest of the cellular architecture of the skin and dermis intact, wound healing did not slow down proportionally, did not take slightly longer. It stalled. The timed sequence of specialized fibroblast types failed to appear in the correct order. The inflammatory phase extended without resolving cleanly into the proliferative phase. The wound remained open chronically in a way that no ordinary variation in healing between individual animals produced. The lineage was not one contributor, it was the sequencer. Remove the architect and the construction doesn't proceed more slowly, it proceeds incorrectly, or not at all. What this means for the tissue that was always cut away and discarded is something that takes a moment to fully register. Every wound that healed in every body that ever lived, healed in part because of cells residing in the fascial layer that the surgical instrument moved aside first. The progenitors that coordinate the repair sequence were in the white membranous tissue that anatomists peeled back to reach the muscle or the organ or the nerve. They were in the layer nobody named for centuries, doing in every wound exactly what the 2023 paper finally described. The differentiation trajectory has been found conserved in human keloid tissue and psoriatic skin bioinformatics datasets, meaning the biology observed in mouse models is not merely a rodent curiosity, but reflects a mechanism operating in human wound pathology as well. In keloids, where scar formation becomes excessive and persistent, the CD201 lineage differentiation may be dysregulated, the myofibroblast gate held open too long, the retinoic acid checkpoint insufficiently maintained. In chronic wounds that fail to heal, the progenitor population may be depleted or unable to receive the signals that initiate differentiation. The wound healing cascade is not a general process driven by generic cells. It is a specific, timed, architecturally precise event driven by a progenitor population that lives in the fascia and differentiates in response to molecular checkpoints set by the wound environment itself into exactly the cell types needed for each successive phase. This is what the tissue that was classified as packaging was doing, in every body, in every wound. Right now in your fascial tissue, beneath the skin of your back, your arms, the soles of your feet, everywhere the connective network runs, CD201 positive progenitor cells are present. They are not activated. No wound has occurred, presumably. They are not differentiating into anything. They are simply resident, maintained in their progenitor state by the ordinary signaling environment of uninjured tissue. They do not need anything to be wrong to exist. They are simply there, as they have always been, in the deepest layer of the connective tissue that holds everything together, the layer that was for centuries the first thing the knife moved out of the way, resident and capable, and waiting in the way that only living things can wait, not expectantly, just present in the dark. You know this feeling probably. Not pain exactly, something blunter than pain. A resistance, the sense that a part of you is moving at a different rate than the rest, dragging slightly as though the tissue underneath has developed an opinion about how far it's willing to go. Might be in the shoulder. It might be the sole of the foot in the first minutes after getting up. It might be somewhere in the mid-back that takes 3 or 4 deliberate movements to persuade into anything resembling its normal range. This is what densification feels like from the inside. The word itself is quiet, unalarming. Densification doesn't sound like a condition. It sounds like something that might happen to a material in a factory, a technicality, a processing step. And yet it describes a state that a significant number of people live with chronically in various regions of the body, without knowing it has a name or a mechanism or anything other than the ordinary frustration of a body that has become less cooperative than it once was. What densification means biologically is this. The loose connective tissue that provides the sliding zone between fascial layers, normally maintained as a hydrated low-viscosity gel by the hyaluronic acid secreted by fasciocytes, has changed its properties. The hyaluronic acid has thickened, its molecular weight has dropped, its water-binding capacity has reduced, and without the hydrated film that allows adjacent fascial layers to glide past each other, those layers begin to adhere, not through scar tissue initially, just through altered gel chemistry, through a lubricant that has become incrementally less lubricating. The causes are multiple and not always separable from each other. Overuse syndromes, the sustained repetitive loading of a particular tissue region, can trigger local inflammatory responses that alter hyaluronic acid chemistry, shifting it toward the lower molecular weight forms that aggregate more readily. Trauma does the same more acutely. Surgery introduces a disruption to the mechanical environment that the fasciocytes haven't been prepared for, changing the tissue architecture in ways that affect how the gel layers reform. Even sustained postural loading, hours at a desk, a sleeping position held too long in one orientation, can over time shift the mechanical environment of the fasciocytes sufficiently to change what they produce and how it behaves. The nociceptors in densified tissue are in a different mechanical environment than they were built for. This is the sentence the pain explanation rests on. It isn't that the nerve endings are damaged. It isn't that there is tissue tearing or joint erosion or any of the structural pathologies that conventional imaging looks for and sometimes doesn't find. It's that the free nerve endings threaded through the facial layers, the same sensory apparatus that detects load and stretch and chemical change, are now embedded in tissue under different tension than normal. The layers that should be sliding relative to each other are instead transmitting force directly across the interface. The mechanical signal arriving at the nerve endings is altered, and the nervous system, receiving an altered signal that it cannot attribute to a specific structural event, registers it as the particular diffuse, difficult-to-locate ache of facial pain, burning sometimes, or a dragging quality, or the stiffness that concentrates in one area and resists explanation. Outside, there might be the sound of something settling, the kind of sound a building makes when the temperature drops and the materials contract fractionally. Small, structural, easy to ignore. The clinical presentations of densification are diverse, but the mechanism recurs. In the shoulder capsule, myofibroblast overactivation and fascial thickening contribute to the progressive restriction of adhesive capsulitis, frozen shoulder, where the connective tissue has reorganized around a contracted, thickened state, and the joint it was meant to support has lost much of its range. In the plantar fascia, densification of the connective tissue along the sole of the foot produces the first step pain that is among the most recognizable features of plantar fasciitis. The nociceptors in the plantar fascia registering the altered mechanical tension of a tissue that has lost its normal glide. In post-surgical adhesions, the disruption of fascial layers triggers a healing response that goes beyond restoring interfascial mobility. New connective tissue bridges form between previously separate layers, developing their own vascularization and their own innervation, becoming autonomous tissue with properties distinct from the surrounding structures. Adhesions are harder to reverse than densification. By the time a surgical adhesion has developed its own blood supply and its own nerve fibers, it has become, in a functional sense, a new structure, one that wasn't there before and that now participates in the mechanical environment of the surrounding tissue in ways the original fascial architecture wasn't designed to accommodate. The pain of post-surgical adhesion syndromes, which affects a meaningful proportion of people after abdominal, pelvic, and orthopedic surgeries, has this quality of coming from somewhere that doesn't quite correspond to the original surgery site, because the adhesion tissue has developed its own sensory apparatus. The reversibility of densification depends significantly on how far it has progressed. Early densification, thickened gel, reduced glide, altered nerve-ending environment responds to the kinds of mechanical input that stimulate fascia site activity and restore hyaluronic acid turnover. The tangential oscillation and perpendicular vibration that mathematical models showed to be most effective at redistributing hyaluronic acid within fascial layers. Sustained movement through full ranges. The load and unload cycles that the fascia site interprets as the signal to produce. These things help measurably when the architecture of the tissue itself remains intact. Later densification, where collagen has reorganized, where myofibroblast activity has elevated local tension, where the interfascial space has begun to consolidate, is slower to respond, and true adhesion, with its new vascularization and innervation, may not respond at all to approaches that work earlier in the process. The window matters, in other words. Not in a frightening way. Most densification doesn't progress to adhesion, and most people's bodies spend most of their time in the middle range of the spectrum, moving between better and worse days, between freer and more restricted, without the process ever reaching the irreversible end of the continuum. But the body is telling something, in the language of restriction and altered sensation, and the telling is worth attending to. Densification is not a failure of the body. It is a biological process, adaptive in its origins, conservative in its logic, that has extended further than is mechanically useful. The gel thickened because the tissue was stressed, or inflamed or immobilized, because that is what the gel does under those conditions, because it has always done that, because the tissue was built to respond to its mechanical environment, and it was doing exactly that. The sliding stopped because something in the environment of the tissue changed. And right now, wherever densification exists in your body, if it does in whatever region at whatever stage, the tissue is not static. The fascia sites are still present, still capable of producing hyaluronic acid when the right mechanical signals arrive. The fibroblasts are still cycling. The interfascial glide, however reduced, has not reached zero. The system is slower than it was, not finished. And somewhere in the loose connective tissue between your fascial layers, in the dark and the warmth of a body at rest, the gel that allows everything to slide is whatever it currently is, thinner or thicker, more or less viscous, at the particular point in its own slow chemistry that it has reached tonight. Present, still in the space between one layer and the next, doing what it can with what the tissue has given it to work with, which has always been enough or close enough to get you here. Before your organs had taken their final shapes, your fascia was already there. This is not a figure of speech. It is the sequence of events. In the third week of human embryonic development, when the embryo is a structure smaller than a grain of rice organized into 3 primary tissue layers, the middle layer, the mesoderm, begins to differentiate. And what it differentiates into, among many things, is connective tissue. The loose and dense fibrous tissue that will become the fascial network of the adult body begins its organization here, in the earliest weeks, before the heart has its final form, before the liver has begun its particular folding, before the nervous system has extended its long reach into the periphery. The fascia is not assembled afterward, like scaffolding erected around a completed structure. It is part of the original construction. The space into which everything else grows is, in large part, a fascial space, organized, bounded, and prepared by connective tissue that arrived before what it would eventually hold. The mesoderm is divided into 3 broad regions during early development: the paraxial mesoderm, which runs alongside the developing neural tube and gives rise to the segmented somites that will become vertebrae and associated musculature; the intermediate mesoderm, which generates the urogenital system; and the lateral plate mesoderm, which splits into 2 layers separated by a fluid-filled cavity and generates most of the connective tissue of the body wall, the limbs, and the visceral organs. It is primarily from the lateral plate mesoderm that the fascial system derives. The fibroblasts, the loose connective tissue, the dense fibrous sheets that will eventually envelop muscles and organs and bones and nerves. This differentiation is not a rapid event. It unfolds across weeks, the connective tissue laying down its architecture gradually, establishing the spatial relationships that the organs will later inhabit. The liver doesn't simply appear in the abdomen. It grows into a space that already has fascial boundaries, a connective tissue environment that shapes its development, constrains its position, and eventually provides the fibrous capsule that holds it together and the septa that divide it into functional units. The same is true of the lung, which grows into plural fascial layers, the heart, which is surrounded by the pericardium, the kidney, the spleen, the gut. Each inhabits a fascial envelope that was organized before the organ reached its final form. There is something the developmental biologist would call organ dependency here. The organs don't merely borrow the fascia's structural support, they are, in a more fundamental sense, dependent on it. Research reviewed in the American Journal of Physiology in 2024 describes this dependency specifically: how the fascial system regulates organ information, pain sensation, and fibrotic processes, how fascial fibroblasts communicate with the microenvironments of adjacent organs in ways that influence disease progression. The fascia is not the organ's container. It is part of the organ's operating environment. Something is faintly audible in the room: the low mechanical sound of something running, or the absence of sound pressing slightly against the ears. The body nearby, horizontal and warm, is completely unaware of any of this, which is appropriate. Origin doesn't require awareness to persist. The meninges offer an interesting edge case. The layers of connective tissue surrounding the brain and spinal cord, the dura mater, arachnoid, and pia mater, are considered by some researchers to be specialized fascia, a view supported by their mesodermal origin and their histological similarity to fascial tissue elsewhere in the body. If this framing holds, then the fascial system extends even into the housing of the nervous system itself, the tissue that was classified as unimportant wraps. In some sense, the organ that decided it was unimportant. I find this detail difficult to pass over without noting. The continuity of the fascial network across the adult body, the way it runs without interruption from the plantar surface of the foot to the base of the skull, from the superficial layer just beneath the skin to the visceral fascia around the deepest organs, is not an accident of anatomy. It is the continuation of an organizational principle that was established in the embryo. The network is continuous because it was laid down continuously as a single differentiating tissue that never stopped being itself, even as everything around it became something specific. What's strange to sit with intellectually, and I find it genuinely strange, not in a performed way, is that the fascial tissue in your body right now is, in a biological sense, the same tissue that organized the space you grew into. Not the same cells, obviously. Cells have died and been replaced many times over across a lifespan, but the same lineage, the same architectural logic, the same mesodermal origin expressing itself through the same family of fibroblasts and fasciocytes and extracellular matrix proteins that have been maintained in continuous biological succession since the embryonic period. The collagen in your thoracolumbar fascia tonight was not there 30 years ago. The cells that made it may have been there 5 years ago, or 2, or may have divided from a parent cell last year. But the tissue type, the organizational identity of the structure, has been continuous. The fascia has never not been fascia, it has never transformed into something else and then returned. It has simply persisted, producing itself forward in time, maintaining the architecture that was first established before you had a face. This kind of continuity is different from the continuity of a bone, which remodels but retains its fundamental shape. The fascial network has no fixed shape. It is tensional, responsive, viscoelastic, distributed. Its continuity is not geometric. It is organizational. The capacity to surround, to connect, to transmit force, to lubricate, to sense. These functions have been running without interruption since before there were organs that needed surrounding. Your body, in this specific sense, has never started from scratch. It began with a connective tissue architecture and has been building within that architecture ever since. Every organ that differentiated into a space the fascia had already prepared. Every muscle that formed within a fascial envelope that predated it. Every nerve that extended its reach into the periphery did so through corridors of loose connective tissue that were already there, already organized, already part of a network that was doing something before the nerve arrived. The fascia, in this reading, is not part of the body's infrastructure. It is the first infrastructure. And right now, in the darkness and warmth of wherever you are, that first infrastructure is still present, still organizing. The same functional tissue that established the spatial logic of your development is still encasing your muscles, still surrounding your organs, still threaded through with nerve endings and lubricating cells and progenitor populations that have been there in some form of continuous biological succession since the lateral plate mesoderm began to differentiate in the 3rd week of a development that eventually, across months and years, became you. The fascia doesn't know this. It doesn't hold the memory the way a mind holds memory, as something that can be retrieved, examined, and lost. It holds it the way tissue holds anything, structurally, chemically, in the arrangement of fibers and the populations of cells and the particular composition of a matrix that has been producing itself forward in time since before you were born. It has always been the first thing. It is still the first thing in some sense, the organizing tissue, the preliminary architecture, the connective context within which everything else in you continues to function, tonight, in the quiet, in the dark, exactly as it has since the beginning. You are being held right now, not by anything external, by the tissue itself, the fascia beneath your skin, around your muscles, enveloping your organs, threaded between every structure that makes you a continuous body rather than a collection of separate parts. It is under tension right now, distributed tension, the kind that gives without breaking and absorbs without collapsing. You don't feel it as pressure. You don't feel it as constraint. You feel it, if you feel it at all, as the background fact of having a body, the sense that everything is roughly where it should be, that the parts are in some kind of coherent relationship with each other, that you are, despite being made of dozens of tissues and hundreds of structures and trillions of cells, a single thing. That coherence is not accidental. It is maintained. The fascia maintains it not through rigidity, but through something closer to distributed negotiation, a continuous adjustment of tension across the network, accommodating the loads that arrive from movement and gravity, and the simple fact of existing in a position, routing those loads outward through the connective tissue so that no single structure bears more than its share for long. When you shift in your sleep, as you will, as you already have tonight, the small unconscious repositioning that happens without waking, the fascial network redistributes. The tension pattern changes across the whole system within moments. The body settles into a new configuration that is, again, held. This has been happening your entire life. There is no moment in your history when the fascia was not doing this. Not in childhood, when the tissue was younger and more compliant, and the crosslinks were fewer, and the interfascial glide was easier. Not during the years when you were growing and the proportions of your body changed faster than you could track. Not during illness, or injury, or the long recoveries that follow certain kinds of damage. Not during sleep, not during the unconscious hours, not during any of the time you have spent not thinking about connective tissue, which is essentially all of your time. The support was running in the background. What I find myself coming back to at the end of all of this, the cells and the gel and the nerve endings and the progenitors and the embryonic origin and the centuries of neglect, is not any single discovery. It is something more ordinary than discovery. It is the quality of the support itself, the fact that it holds without gripping, that it gives under load and absorbs what it can and transmits what it can't, that it responds to movement without requiring conscious direction, and that it maintains itself largely with the materials and signals the body provides in the ordinary course of being alive. This is not a small thing to do. It is, in some ways, the central thing, the organizing function without which nothing else in the body has the spatial context it needs to work. The muscles need connective tissue envelopes to define their boundaries and coordinate their force. The organs need fascial envelopes to maintain their position and participate in the sensory environment of the body. The nerves travel through corridors of loose connective tissue. The blood vessels are wrapped in it. Everything that moves through the body, everything that holds still in the body, everything that senses or transmits or repairs or lubricates, all of it does so within a fascial context that was there first and has been there continuously. The second skeleton of the title is not quite right, actually. A skeleton implies rigidity, a fixed frame, a structure that holds things in place by refusing to move. Fascia is almost the opposite of that. It holds things in place by moving with them, by distributing, by giving where giving is appropriate and transmitting where transmission is needed, and absorbing where neither of those options is sufficient. It is a holding that is always slightly in motion, always adjusting, always negotiating with the forces that arrive and the structures that generate them. You carry this. You have always carried it. It has never been separate from you, never been a system you could step outside of and examine, never been anything other than the immediate context of everything you do and feel and sense. Somewhere in the loose connective tissue between the layers of your thoracolumbar fascia, a fasciocyte is producing hyaluronic acid. In the progenitor population of your deepest fascial layers, CD201 positive cells are resident and quiet. In the collagen architecture of your deep fascia, fibers laid down over years of loading and repair are holding their arrangement. 250 million nerve endings are reading the mechanical state of the tissue around them, routing most of what they find away from consciousness, because none of it requires your attention tonight. The system is running. If tonight's chapter found its way to you, if something in the biology stayed with you or settled something or just made the body feel less opaque than it sometimes does, then leaving a like is a quiet way of telling me that. Not because the number matters, but because it helps this particular corner of the internet stay findable by the next person lying in the dark wondering what their body is made of. And if you've found yourself here more than once, If the sleepy biologist has become part of how you end your nights, then subscribing is just making that habit official. There are people who support this channel through membership and through super thanks, and I want to say, without making too much of it, that those people are the reason this space exists as something I can keep returning to. If you've ever been one of them, thank you. If you're curious about it, it's there. And if you have a thought about fascia, about the body, about something you've always wondered and haven't found a quiet enough place to wonder it, the comments are open. They are genuinely one of the parts of this I find most worth checking. Outside, the night is at whatever hour it has reached. The room has whatever temperature it has settled into. The sounds, if there are any, are whatever sounds belong to this particular place at this particular time. Your body is horizontal. The fascia is doing what the fascia does. The gel between your layers is whatever viscosity it has arrived at tonight. Thin enough probably to allow some gliding, enough to let the layers move when you next shift position. The nerve endings are quiet or nearly so. The progenitors are present. The collagen is holding its arrangement in the dark. And the tissue that was discarded for centuries, cut through, set aside, unnamed, classified as the wrapping rather than the thing worth keeping, is doing right now in the warm interior of a body at rest what it has always done, holding everything together, without gripping, without announcement, in the continuous unhurried way of something that has never needed anyone to notice it in order to keep working, and that will still be working quietly when you wake.